Today's practical is on decay, which is one of the required practicals for biology. In this practical, we will be investigating the effect of temperature on the rate of decay of fresh milk by measuring a change in pH. Milk left at room temperature decays very slowly, so we won't be able to observe that in the time we have to do the practical in. So we will be modelling decay by using the help of an enzyme called lipase. Lipase will break down the fatty molecules in the milk to produce acidic substances. Now, in natural decay, the fall in pH is due to the production of lactic acid. But in this demonstration, the fall in pH will be due to the production of fatty acids as a result of the enzyme action. The hypothesis of this practical is that as we increase the temperature, we increase the rate of decay because the microorganisms work faster and better at higher temperatures. The independent variable in this practical is temperature. The dependent variable is the time taken for the solution to turn acidic. And the control variables are the amount of solutions used, the age and type of milk. For this practical, we will need a water bath, a thermometer, a stopwatch, lipase in a solution, full fat milk, sodium carbonate solution, cresol red indicator, and 2 test tubes. One test tube should be labelled milk and the other test tube labelled lipase. So first we need to transfer 5 centimeter cube of lipase to the test tube that we have labelled lipase, which I have already measured out here using a syringe. So I shall put that into the test tube now. Now, the second test tube, which I have labelled milk, in that first goes some cresol red indicator, and we need about 5 drops. I shall measure the cresol red using a dropper pipette because we have to be exact with the amount we use. 1, 2, 3, 4, 5. Now next, I need to add in 7 centimeter cubes of sodium carbonate solution, which I have already measured out in this measuring cylinder. So in that goes. And finally, we need to add in 5 centimeter cubes of the milk. Okay, as you can see, the cresol red indicator has changed the milk to a pinky purple colour. That indicates alkaline conditions. Now we need to prepare our water bath so that it is at a temperature of 45 degrees Celsius. And we can usually prepare this by taking some water from the tap and then topping it up with some hot water. I have now just placed a thermometer into the water bath so that I can measure its temperature. The temperature is reading exactly 45 degrees Celsius. I will keep the thermometer inside the water bath so that I can monitor the temperature throughout the practical. Next, we can add in both of the test tubes to the water bath. And we need to wait a short while for the contents of the test tubes to be the same as the temperature in the water bath. This can take a short while. The temperature of the water bath is still at 45 degrees Celsius, and the temperature of the contents should roughly be now the same as the water bath. I will now transfer 1 centimeter cubes of the lipase solution to the milk test tube using a dropper pipette. And at the same time, I need to start the stopwatch because I need to time how long it takes for the milk to become acidic. The creasel red indicator will turn the milk to a yellow colour when this happens. Just give it a gentle stir so that the lipase mixes well into the milk. This can often take a short while. I have now stopped timing because the milk has turned to a creamy yellow colour. This indicates that the milk has become acidic now. We need to repeat this step so that we can obtain 3 measurements, as we need to calculate a mean value. And we need to calculate a mean value to obtain accurate and reliable results. And also if we were to plot a graph. This step can be repeated with various temperatures, either higher or lower than the one which we have investigated with now. Results can be used to plot a graph. We investigated with temperatures 20, 35, 45, 50 and 60 degrees Celsius. Our results show that at the low temperatures, such as 20 degrees Celsius, the mean reaction time was 359 seconds, whereas the reaction at 35 degrees Celsius took 24 seconds. And at 45 degrees Celsius, it took 20 seconds. This shows that the reaction was much slower at 20 degrees Celsius, and that's because the lipase works slowly at low temperatures. Now, at higher temperatures, such as 50 and 60 degrees Celsius, the reaction has slowed down or possibly has stopped, as the milk did not change in colour even after 5 minutes, indicating that the enzyme has denatured. We can see that the reaction is at its fastest at 45 degrees Celsius, because this is approximately the optimum temperature for lipase. A graph can be plotted with the temperature on the x-axis and the time taken for the milk to turn yellow on the y-axis. We can see from the graph where the reaction was the fastest and the slowest. We can plot a second graph with the temperature on the x-axis and the rate of reaction on the y-axis. We can work out the rate of reaction by dividing 1 by the time taken in seconds for the milk to turn acidic.